Hello, everyone, and thank you for joining our webinar. Before we get to our featured talk, I want to give you some background on Labcyte and our products. Labcyte invented the use of focused sound energy to transfer liquids and began shipping our flagship product, the Echo Liquid Handler, in 2004. Initially, the Echo was used by large pharmaceutical companies for high-throughput screening and sample management. But over time, more areas of research became dependent on Echo liquid handlers for high-performance liquid handling and transfer of a wide range of fluid types, sample types, and reagents. Also, over time, we've developed complementary product lines to really help implement the Echo into a number of different workflows. We have a robotic integration platform we call the Access Workstation, and this is used to integrate the Echo liquid handler with a variety of devices and to provide walk-away, fully automated processing using the Echo system. We also provide and offer pre-qualified Echo source plates, and these plates are tested to make sure that every transfer of fluid from the Echo liquid handler is highly accurate and precise. We also have a line of software products, a suite of Echo software applications, and these software applications are tailored for specific laboratory workflows, and they provide a graphical interface to set up protocols for the Echo and also simulate and kind of troubleshoot protocols before you actually run them on the Echo system. We offer two models of the Echo liquid handler. Our 525 model transfers fluids in increments of 25 nanoliters, and our 550 and 555 models transfer in increments of 2.5 nanoliters. We initially produced the 550 and 555 systems to target high-throughput screening, where our users wanted to transfer as little compound as possible into their assays and screens and really conserve their compound libraries. However, as I mentioned, the Echo spread into a lot of other research areas. And in some of those biology labs and core labs, we realized that it wasn't miniaturization that our users were after, right? They just wanted that high accuracy and precision that they could get from an Echo system. And there, when they were working with larger volumes, they found that 2.5 nanoliter increment to be too small. It took too long to build up the larger volumes that they were looking for. So we developed the Echo 525 liquid handler with a 25 nanoliter droplet. So it's 10 times larger increment that you can use with that system. And it builds those larger transfer volumes much faster. And so this next slide kind of shows the technology inside the Echo. At the bottom of the slide, you can kind of see an acoustic transducer. Now this transducer emits sound energy through the bottom of the microplate well and up to the meniscus of the fluid. And that bottom plate well, if it's damaged or has some distortion, it could affect the transfer. And that's why it's very important, and we recommend using our pre-qualified source plates to avoid that. Nonetheless, that sound energy, if it passes through the bottom and goes up to the meniscus, it's focused at the meniscus, and it starts to create enough energy there to actually eject a droplet of liquid upward into an inverted destination plate. So that drop travels up. Because we're controlling the power that's being applied to the meniscus, that drop is consistent, drop after drop, and the drops come out very fast, up to 500 droplets per second. So you can build up large transfer volumes very quickly. And, a lot of people look at the inverted destination plate and worry about fluid slipping out or leaking. And that, because of the capillary forces within the well, that doesn't happen. And again, Echoes have been used for a variety of reagents and a variety of applications, and there hasn't been any dripping issues. What's really nice about the technology as well, that transducer can move from well to well. and there's no use of any kind of tip. So you can quickly transfer fluid from one well and move to the next well without doing any tip changing or any kind of tip washing or anything like that. Also, because no tips are used, there's no contact with the fluid at all. It's completely non-contact. And there's no risk of the fluid sticking to the tip and getting carried over from one well to another. So you don't have to worry about things like sample carryover, cross-contamination. And also, I mean, you get to save costs on tips as well. We also have a very cool technology inside the Echo called dynamic fluid analysis, or DFA as we call it in a lot. With this, we're using sound to profile the well. So in the slide before I talked about building up energy at the meniscus and ejecting a droplet. This we use a slightly lower power, so we don't eject a droplet, but we can kind of send sound waves up, and as they bounce off the meniscus, collect information from them. And so with this, we can see where the meniscus is, so we can tell the overall height of fluid in a well, and we can check the speed of sound and understand how viscous the fluid is. And this allows us to adjust on the fly, right? If you go from one fluid type to another and the viscosity changes, the Echo can actually sense that and in milliseconds adjust power to ensure that you get your 2.5 or 25 nanoliter droplet. What's really nice about that is if you're working with fluids that can hydrate or evaporate, can change while you're transferring across a plate, the Echo is adjusting with every transfer as it goes to, again, assure that those droplets are the appropriate volume that you want. And again, getting to very highly precise, highly reliable liquid transfer. And I should mention that the Echo, again, has been used across a number of different fluid types, right? So transferring cell lysates, DNA, RNA, different types of assay reagents, and of course, DMSO samples. So this example is kind of an example of what's done in synthetic biology for DNA assembly. So you can see the microplate on the left has a number of different colored rows. Those can represent different oligos that you'd want to pool into an assembly reaction, or a combination of oligos and the actual assembly reagents. And the Echo can transfer from any one of those wells at any time and pool them into a plate. And again, because there's no changing of tips or washing of tips, that pooling happens very quickly. And the instant feedback we get from a lot of our users, especially working in the synthetic biology space, is that the system saves them so much time immediately. And that's in addition to any miniaturization and cost savings from miniaturization. This next slide kind of highlights that in a kind of a simplified infographic, right? The system at the top is to represent kind of a tip-based liquid handling system or traditional liquid handling system, and then the bottom section is the Echo liquid handler. And if you look at just the assembly portion, you're already saving around 9 hours. Now, this is going to vary lab to lab, but assume you're cranking through numbers of assembly reactions, not having to change all those tips and change your pipetting technique every time you transfer can really save a lot of time. Additionally, a lot of groups, once they've assembled their genes, they'll go through QC steps, sometimes using sequencing, next-generation sequencing. And there, you typically build your sequencing libraries and then pool those libraries and normalize them. And with the Echo system, because you can transfer low volumes of high concentration and larger volumes of low concentration, all without changing tips or doing any kind of dilutions, you really save a lot of time. You can quickly pool and normalize in the same step. And you combine those two together, and we have users who are saving around 15 hours in that whole process from just those two steps. Also, the Echo can be used in a lot of other steps as well, right? So if you look at functional testing, you can use the Echo there. And, if you're looking at transfection, the Echo can be used there. So really, the echo can provide a benefit across the entire set of workflows that are typically used in synthetic biology. And as I mentioned, miniaturization is also another big benefit. And, you know, we've kind of worked with lots of different research areas over the years and kind of collected these examples of where our users have been able to take standard methods and reduce the volumes, right? If you look at qPCR, PCR, they've had a 6-fold reduction. We've had users go to a 100-fold reduction for our next-gen library, and these are, you know, published results that we can circulate and show. And as you can see, it has a significant impact, right? You're saving a lot of cost by simply implementing the echo, in addition to benefits of speed and you know, higher precision, higher accuracy from your liquid handling. And as I mentioned, you're not using tips to do this, right? So there's no contact with the fluid. There's no cross-contamination. So that's hopefully a good overview. I'd like to turn it back to Lynn. And again, thank you all.
Thanks for tuning in to this webinar. My name is David McClymont. I'm the Head of Automation at the London DNA Foundry, and we're based at Imperial College London. And today I want to talk to you about the high-throughput DNA assembly methods that I've developed here using the Labcyte access systems. So there's me. So just to give you an overview of what we're going to talk about, you know, I'll give you an introduction to synthetic biology, and, you know, some of the work that's been done in that area and some of the things this field's trying to achieve. And, you know, I'll introduce you to, you know, this idea of the design, build, test cycle. So, you know, what we've been doing with these systems is bringing together this idea that you can start to design genetic circuits and then have these automation platforms build them. And, you know, what kind of conclusions I've came to is that this is both a combination of, you know, traditional assay development, high-throughput screening type stuff, but I feel that, you know, a lot of the times now when we're working on these build procedures, that software development becomes an integral part of that process too. And by having the assay development and the software development happen together, I feel it can lead to an optimum solution. And by doing these things together, I think it pushes the capabilities of what the Labcyte access platform can do. So here's a slide I like to use to introduce the potential of synthetic biology. So this is an up-to-date phylogenetic tree of all the different types of organisms that are around. So in the corner, you can see, you know, eukaryotes. Humans are away in the bottom right there. But you can see that this gigantic tree of life, you know, has been developing over, you know, millions upon millions of years. And these organisms have developed metabolic pathways and enzymes that could be useful to us. And, you know, by using their DNA sequence, we can start to put those bits of information together and start to use them to engineer biology rather than try and understand it. And what really fascinates me is the organisms that are marked in red actually have never been seen, never been cultured. So they're only present and known about by their sequence, which really shows the potential of what we can do, and we can start to write out DNA and start to use these written DNA parts and put them together in certain ways to exploit the engineering principles and apply that to biology. So, you know, here's another slide to kind of introduce the concept. So this is synthetic biology visual, SBOL visual, and what it does is it abstracts away the type of different genetic elements that you can put together. So you've got promoters, which, you know, initiate expression, you've got ribosome entry sites, you've got terminators, and what they've done with this is they've taken the principle from electronics to put together symbols. And once you start to have symbols for things, then you can think about design. So if you want to design some sort of coding sequence, some sort of enzymatic pathway, by having this graphical representation, you can start to abstract that away and really understand and develop new designs, and then lead into the further problem of how you actually build them. So, you know, what is the goals of synthetic biology? So if we take these designs, you know, and we take these sequences that we can get from various databases, we print them out and we can put them together in different ways, but also we want to do that for a reason. We're bringing engineering principles to biology, you know, from bottom up, trying to make things and do useful things with biology. So that can have a range of different uses, from industrial enzymes, production of chemicals, fuels, biofuels is obviously a big area, vaccines, antibodies, pharmaceuticals, genetic circuits, or even data storage. By using the genetic elements, you can start to bring together these principles and start to engineer things you want to do. And that's what we've been trying to do at the London DNA Foundry. So here's a slide here. So Jeff Baldwin and Marco Storch at Imperial College came up with what I think is a neat idea for DNA assembly. So what they did is they kind of standardized prefix and suffix sequences, which can then be added to plasmids in order to create these genetic elements, these polycistronic units. And by taking parts and by taking prefix and suffixes and knowing how they come together with their complementary DNA overhangs, you can bring them together in different ways. You can change the order, you can change the strength of things like different types of promoters, or you can change the strength of ribosome binding sites. And by having this simple logic in a similar way to, fundamental electronics was, we can start to then build up bigger and bigger constructs with lots of variation from as few parts as possible. And that's why I feel the real strength of this is because the variation comes from the prefix and suffix linkers and not from the genetic elements, the plasmids, the coding sequences themselves. So for one coding sequence, you can have it in any order, in any position with any ribosome binding site, and that all comes from one gene. So when you're going for your synthesis provider, you can buy one gene, but you get all the variation through the prefix and suffix linkers. So, how do we approach this? Build process development. Like I said, assay development is, high throughput screening, 384 well plates. It's something people have been doing for, quite a long time. You've got robotic work cells like the Labcyte Access, small focused work cells with different types of capabilities. But where I think the missing link here to do these kind of build processes, I think, is software development. And I think that software development approach feeds back into the assay development and how you approach the design of work cells. And only by taking into consideration all these things together, I think now is the only way to effectively do high-throughput DNA assembly for synthetic biology. So I call this a kind of agile development approach. By considering the assay development, software development, and automation solutions together, you can replan and optimize what you do, because one solution that might seem sensible in your software approach might not work within your assay. So by using scripting, you can quickly develop different approaches together with developing different approaches in the microplates. And, as you do that in the microplates, you can set benchmarks, you can quickly deal with potential showstoppers, and if there's a problem, then you can go back and change your mind and think about a different approach, and then go back to your software and think about how that would work with your different approach. And by having the London DNA Foundry, we've got multiple different work cells with multiple different liquid handling capabilities. We can come up with multiple solutions even across the work cell platforms. So I found this is quite a strong approach, and has been very useful in building these DNA pathways that we'll be talking about. So here is the London DNA Foundry. So, just to give you an idea of what we've got, we've split it out into different work cells. We've got the Labcyte Access on the right, where we've got the 550 and the 525. We've got various types of tip-based liquid handlers from SciBio, and they're located in two work cells on the left. The idea here is I'm just going to show you, how we set this out. We don't want a large system. We want, redundancy, and we want the ability to, have multiple solutions across different work cells. And by having multiple different solutions across multiple work cells gives us more choices on how we do the approach. So, I think this agile approach can also apply to assay development. And I think over the years I've taken that into consideration too, because if someone brings you a protocol and you start to break it down to its constituent parts, by breaking it down to its constituent parts, you can start to optimize how you do that. So you might find that one of those steps is not required. You might find that one of those steps require the integration of something like a centrifuge. But if that's the case, rather than try and integrate that instrument beforehand, you can then always just use a standalone centrifuge and really see if that's a bottleneck that has to be met. And also by breaking it down into its constituent parts, You might be able to say, well, this is a simple step or this is an easy step. I can prioritize that, or maybe it's a difficult step, so I can work on that much more quickly. So here's an idea I came across from working on a previous project, and what I was trying to do here was work on a project to do with personalized medicine, and we had a whole set of different compounds we wanted to dispense over different concentration ranges that sometimes we may have wanted to do combinations, or we may have wanted to do different subsets of that. And what I realized is that if you could define what your experiment in your own way, and you could build some sort of interface that absolutely met your own experimental paradigm, you could generate these pick lists automatically and run them on the Access platform fairly easily. So, your experimental paradigm, what you're trying to do, really what you're trying to focus on, everything can become a pick list at the end of that process. And if it's a pick list, and if it's got a barcode on it, and if it's got a transfer volume, the cherry pick software together with Tempo can really crank through those dispenses quite fast without failing. And, by having these pick lists, they can be, of up to 10, 20, 50,000 lines, the Tempo software is very well suited to just running through those lists. So if we come back to synthetic biology, what were we trying to do here? So here's synthetic biology visual language there. So here is a CAD operon. So we've got a promoter, a ribosome binding site, we've got a coding sequence, a second ribosome binding site, and then another coding sequence. And, as an example, maybe that's what we would want to build. But also what we wanted to do is we wanted to introduce design of experiments at the very beginning. So we have, jump software to help do that. And these design of experiments can then give us sets of promoters and coding sequences, and then automatically gives us a balanced statistical model to work with. So as another principle, what I wanted to do was to have some sort of software generate these build instructions, which are essentially cherry pick lists for the robot, for unlimited number of designs for any size of design. And I wanted to do that in the optimum way by considering the assay development wetware approach and the software solution at the same time. So, assay development. So, we talked about how the basic assembly system worked. And what I think is that small experiments can really influence the overall approach. So I've been using the Echo to develop the assay in the microplates. You can quickly do that with the plate reformat software and cherry pick, and you can devise experiments for go no-go decisions. So on the top left, we have this digestion ligation reaction. So what we can do is we can check the sizing. Have we optimized that in the 384-well PCR plates? Can we ligate these linker sequences onto the part we're interested in? And the fragment analyzer gel information there can show that's possible. The second thing we wanted to work with was, magnetic beads. So the magnetic beads can remove these free linkers because they are oligos with complementary overhangs. They can, reduce the efficiency of the downstream assembly. So this gel here, if you see below the 35 base pair line, the one on the right shows that we can, effectively remove the magnetic beads. And then as the last experiment here is, can we have consistency across the plate? Not only does that show us reproducibility of our reactions that we've optimized on the Echo, but it also ensures that it's consistent across the plate. And it's reproducible. And as these experiments lead to go, no-go decisions, these can actually lead to your downstream QC, because as you start to do, thousands and thousands of these as high throughput builds, the downstream process QC is incredibly important. So, how do we come to this process from start to finish? As we kind of optimize the experimental conditions for the digestion ligation, we started to think how that would work as an end-to-end process. So we have parts, and we want to have high volumes of those parts. So we look at tubes and 96-well plates. We want to bring those into Echo LDV plates for the digestion ligation, and you can see the optimized reaction there. We want to come out of the 384-well plate back into 96-well plates for magnetic bead purification. And then we want to come back in for another echo pick list to do what we call the stitch reaction, which puts all these together in the correct order. So we've got a number of different pick lists coming in and out of 384 well plates, coming in and out of the echo systems. But from start to finish, we can manage this process effectively. So we talked about, the assay development and the process development, but what we wanted to bring to the forefront of what we were doing was design of experiments. Because as synthetic biology is a fundamentally engineering discipline, the DOE approach we felt had to be brought in from the start. And we used jump from SAS, and what we can do is we can have a custom designer, which then tells us what experiments we need to do. You can see that example on the right. And as it tells us to do these experiments, it's building that statistical model underneath, but it's also making sure that's balanced for the number of different factors that we're trying to test. And I was really interested in how I could merge my software approach with my build approach and then automatically generate the instructions to do that based on the DOE as well as all the other factors that we were testing. If we think about, again, what would the application look like for this? So on the left-hand side, we have jump. We have, the scripting capability. And also we wanted to have that context in our LIMS system. So by mixing the two together, the scripts and jump, and using the context of what type of parts we were interested in as defined by the DOE in the LIMS, we could start to generate algorithmically the pick list required to actually build these. And, by, using this agile approach, by using the scripting, we can then run cherry picks of, multiple sizes and execute them on tempo quite easily, similar in a way to the approach I took with the personalized medicine system. Once this development approach has been completed, we can, we've got QC and we've optimized everything. We've produced a robust assay. We've got the iterative development of both the software and the wetware to develop the minimum viable product that meets all the requirements. And then after that, we can build that out. Once we can move on to building out that process, we can settle on standard operating procedures. We can settle on how the system integrates with the work cell through Tempo, and then we can embark on a project to do the software engineering required to make that a scalable process that scientists can use. So here is my software. So what this software does is it will take your design, it will merge it with your design of experiments, and then in the background, what it will do is it will generate and figure out the way to build it. So it will figure out the way to build it across a number of different assays. And what I realized was that as the number of these assays was quite high, my worry was that, the scientists running these projects would need a dashboard or some way to manage that. So in here, this experiment will take that design and then generate all the pick lists required. So here you can see the pick list for parts of the process. We've got the job master, which is getting the parts out of the storage. We've got the clip reaction, which is digestion ligation. And we've got the stitch reaction there, which is the final one for bringing all these things together. And by having all these in one place and having them all generate barcodes and generate the pick list, I feel this is an effective way to manage what could have been an unwieldy approach, because there's so many pick lists and so many different assays. I felt that this job dashboard that essentially creates all the build instructions for the Echo platform was a necessary requirement to actually do this effectively. So, one thing, we're talking about the Labcyte equipment here, and what I've found is that it's a really useful platform for doing checking that you don't have genomic DNA contamination. So we have an assay here whereby if there is genomic DNA in your samples, the fluorescence of the quantification will show you if that's there. Now, the genomic DNA can really reduce your DNA assembly efficiency for a number of downstream applications. So we use this as an upfront QC using the Echo system to make sure that any parts that we check into our library have met that QC step of having reduced amounts of genomic DNA. So this figure here will at the top shows you the manual transfer of a set of promoters that we received from a DNA synthesis supplier, and you can see that there's the replicates are generally in the same range. But when we do the DNA quantification assay on the echo, we can see that a number of those promoters either don't transfer at all or have high variability when they transfer. So when you run the ones that don't transfer on an agarose gel, which you can see on the right, you can start to see that correlates completely with genomic DNA contamination. So the ones that I've circled on the right, promoter 15, you can see has a high level of genomic DNA. Promoter 16 has a low level of genomic DNA and hence transferred on the echo fine. And then, beside that, promoter 17, has high genomic DNA, as you can see in the agarose gel, and on the echo you see that it hasn't transferred. And this is really important to ensure the efficiency of your downstream DNA assembly techniques. And the way we do that, we've got an optimized echo assay for that in 384 wells, no intermediate dilution of the pico green. We've got 100 nanoliters of DNA, and we take that up to TE buffer, and that's a really quick assay in 384 well plates that we use routinely to QC all parts coming into our library. So again, and further QC, I think that's really important when you're talking about large-scale automated processes. You have to have the ability to trust what you're doing. And for every single DNA assembly we do, we run everything on the fragment analyzer, check all the sizing is correct, and all the magnetic beads are gone. Similar to the assays that we worked up as we were developing the approach. Now that becomes a standard part of the process to ensure consistency as we build more and more different scales of constructs. So I also think there's a couple of features that I'd like to talk about. Thermocycline is an integral instrument to synthetic biology, and there may or may not be different options that you can look at for your Access platform. But the option that I wanted to look at was the T-Robot. And there was a little-known feature within Tempo that allows you to execute your own program. So if you can execute anything, any piece of software on that system, you can start to integrate your own instruments, and that was, really key for us being able to use this effectively. So I built my own driver for the T-Robot as an executable, and then within Tempo, we can choose the execute program, and then the robot will send the take a plate to that position within the scheduler. And that's been a really, really key thing for us, to not be limited by what was available. By taking that Access platform and extending it out, we've really kind of pushed and enabled us to do things in a much higher throughput. So, what have we achieved when we can do this? Just to put some numbers on it. If we have 1,000 construct assembly, 1,000 different genetic elements, pathways, all defined by design of experiments, and those contain 10 parts per stitch. So a stitch is just a collection of different DNA elements. So what we've done is we've got an automated build, but not only is it automated, it's an optimum build solution. So it's actually self-optimizing and making sure that it produces enough different copies of different things or different ways of joining things together in order to minimize the use of materials. So that 1,000 construct assembly, when it goes through our algorithms, produces 15,000 individual transfers. Now, if you're coming from high throughput screening, 15,000 transfers isn't a lot. That's correct. But what we're talking about here is we're talking about aqueous liquids. We're talking about DNA and TE buffer. We're also talking about levels of multiplexing in different ways. So different parts can come together in different ways, and that happens twice between the clip reaction and the stitch reaction. So that algorithmic optimization, is not just 15,000 one-to-one transfers, it's 15,000 transfers that come together and multiplex in multiple different ways. And through this system, it runs across 14 different plates on 6 where automated processes, different types of assays, across 3 robotic work cell systems that we have, and it absolutely requires two echoes, the 550 and the 525, within our access platform as a main workhorse of what we're doing here, because there is just no way to do that number of cherry picks anyway else, and we can do that in one day. Where does this lead us? Well, the first time I produced this figure, it was fascinating to me because it looked exactly like the sort of thing you would see from high throughput screening. But what we've done here is we've done high throughput synthetic biology, and this is where we want to go with this. By automating the process and automating the build and having that automatically be populated into the design of experiments model, we can start to see we're producing lots and lots of data very easily over a space of time. So this is 459 different constructs, and we're showing different types of fluorescent readout there. But that single point plotting against raw fluorescence data, but because we brought design of experiments into it earlier, there's another element to that. By having the design of experiments lead to the automated build procedure from the very beginning, we feel we can add a lot more value to that information. So, if we talk about, what we're trying to do, we're building a model of what we're doing in the cell. By having these data points defined by DOE, we can actually look at the model. We can look at what the significant factors are, and we can use the prediction profiler to make sure that that model actually works. And this is a lot more value, I think, than the single point data, because we can at a glance see this entire experiment in a few number of figures. So we can see that in the top figure, we can see all the important factors involved in our model and experiment, and in the bottom we can visualize how that looks. And by automatically having that be defined by the DOE, we can really quickly bring those engineering principles into synthetic biology by having DOE at the very start. So in summary, what have we done? We've got synthetic biology build, we've achieved that, and we've done that by bringing software and wetware approaches together across a number of work cells with a range of capabilities, and that's how we designed the London DNA Foundry. So by having this agile approach, we can have go, no-go decisions. We can replan and optimize our solution as we go along. And once that's been decided, we can implement that solution, settle on an SOP, settle on how we interact with things like the Tempo platform, and then build the user-friendly custom software. Because, automated DNA build, although I've just said it's 15,000 transfers, it's not a brute force solution. 15,000 transfers with a number of different multiplexing capability steps there are just not possible by hand. But what we've done here is we've expanded the capabilities of what's possible, and so people can focus on coming up with designs by, completely solving the scale of this by, having an optimum software design approach and have it be automatically run on automation work cells. That whole process that people have been doing in the lab by hand becomes one process. They give us the design and we execute it. And, just up the road is the Design Museum Kensington. And, I think that's, an interesting way to look at this approach now, is that if you abstract all this build away from people, what can they come up with? What new ideas for genetic circuits and engineering biology methods can they do? Because we've got the capability here, and we've, used a number of different approaches to make that optimum solution possible. So, just some acknowledgements. So, I wouldn't be able to do this without the help of a lot of people. Lorna, Chris, Jeff, Marco, Margarita, and Chris. I just want to thank them.